Hello everyone, welcome back. Today I'm here to discuss the eighth part of this questionnaire. In this part, I will cover questions from evolutionary biology, molecular biology, and microbiology. Let's dive in. So the first question for today is: new species developed due to our options are isolation and variation, isolation and competition, mutation and competition, isolation and mutation. So its right answer is isolation and mutation. Isolation means geographic, behavioral, or reproductive isolation. And the other term used for this origin of new species from pre-existing species, this is also known as speciation. Formation of new species from pre-existing species. As we know that these members of the species are reproductively isolated. For example, we have this ancestral species that give rise to new two species which are reproductively isolated. So new species develop through isolation, which may be geographic, reproductive, or behavioral isolation, fostering genetic variation. There are environmental factors and genetic drifts further which diversify isolated populations and leading to the formation of distinct species. Then we have mutation. Mutation, as we know, that this is sudden, large, and inheritable changes which are developed in an organism and are inheritable changes. So they are genetic variations that again leads to reproductive isolation or adaptations, and competition reinforces selection pressures favoring advantage traits and facilitating speciation. Speciation is formation of new species. Lastly, isolation prevents gene flow, allowing mutations to accumulate independently, driving genetic divergence and emergence of new species. So the most important factor for the origin of new species is isolation and mutation. So what is speciation? Speciation, as I have already discussed, that it is the formation of new species from pre-existing species. So speciation is the evolution of one plus species from a single ancestral species. This is single ancestral species will give rise to more than one species, and it occurs when members of population becomes isolated from each other. Once two populations are reproductively isolated, they are considered as separate species. That is called speciation. That is for. The next question is: example of an analogous organ is, options are tuber of potato and sweet potato, tuber of dahlia and sweet potato, and tuber of potato and Jerusalem artichoke, and all of them. So its right answer is tuber of potato and sweet potato. Analogous organs means the organs which look similar but they have different origin. इनका origin different होता है but they have some similarities in their appearance. So as we know that this potato tuber is an underground stem, while this Ipomia batatas, that is sweet potato, it is a modification of a root. So they have different origin, but they show some similarities, and that's why these are known as analogous organs. So potato and sweet potato are examples of analogous organs and not homologous organs, although they have similar functions of storing starch. Potato tubers are modified stem that we already know, while sweet potato tubers are modified roots. Therefore, they differ in their structural origin and are considered as analogous organs. Then we have some differences between homologous and analogous organs. These homologous organs, they differ morphologically, but they have similar origin and have similar internal structure, while these analogous organs, they have superficial resemblance, but they differ in their origin and internal structure. They develop in related organisms, and while analogous organs, they are developed in unrelated organisms. Then in homologous organs, stages in development are similar. They have similar development patterns, while in analogous organs, stages of development patterns are different. Then they perform different function. In case of homologous organs, while analogous organs perform same function that I've already discussed. Homologous organs, they are inherited from common ancestor, while analogous organs are not inherited from the common ancestor. The examples of homologous organ includes wings of bat, then lateral fin of whale, and human arm. In plants, we can say that potato tubers and artichoke, Jerusalem artichoke, these are the example of homologous organs because both of them are stem modifications, while analogous organs, where examples are bats' wings and butterflies' wings. So bat's wing, that is a skin modification, while in butterfly there is not a modification of skin. Both are used to help creature to fly, but they have different patterns in bone structure. So this is the photograph of this Jerusalem artichoke. So Jerusalem artichoke and tuber potatoes, potato tubers are homologous organs because both of them are. Next question is the number of chromosomes in the endosperm of dicot having 12 chromosomes in the megasperm mother cell will be. Options are 42, 24, 8 or 26. As we know that this endosperm is diploid tissue and its chromosome number is 12, while this megaspore mother cell is diploid structure. It's 2 and the basic chromosome number, that is haploid structure, chromosome number, that n is equal to, here it should be 4 and the diploid chromosome number is 8. So its right answer is 8. So this is the central structure, which is endosperm, which is formed after the result of double fertilization, and it is formed by the fusion of two polonuclei with the second male gamete, and the first male gamete fuses with this egg. So the megasperm mother cell is diploid. During double fertilization and angiosperm, one sperm fertilizes the egg that form diploid zygote, while the other sperm fuses with the diploid central cell, also known as polonuclei. They form triploid endosperm. Here the chromosome number is 12. Since the megasperm mother cell has 8 chromosomes, that is 2n is equal to 4, and haploid chromosome number n is equal to 4. The endosperm will have 3 times the haploid chromosome number, that is triploid, so which is 3n is equal to 3 into 4, that is 12. So it means this megaspore mother cell of the dicot plant has 8 chromosomes and the endosperm will have 12 chromosomes. This is the right answer. Next question, cellular structures which disappear during mitosis or meiosis are nucleus and nuclear envelope, plastids, plasmamembrane, none of the above. So its right answer is nucleus and nuclear envelope. So the other structure which gets disappeared during the process of mitosis and meiosis, while this plastid remains there and the plasmamembrane again remains there during the division, which is mitosis or meiosis in plant cells. As we can see here in the interface, there is clear cut the structure, that is nucleus and the nuclear membrane, while in later phases of mitosis, this nuclear membrane gets disappeared and nucleus also gets disappeared. So the cellular structure that disappeared during mitosis or meiosis are nucleus and nuclear envelope. During mitosis and meiosis, the nucleus and nuclear envelope begins to disappear as a part of the process of cell division. And the nucleus where ribosomal RNA process, so this is the site of rRNA synthesis, and the nuclear envelope which surrounds the nucleus disassembled to allow for the proper segregation of genetic material and formation of daughter cells. The plastid and plasma membranes are not structures that disappear during mitosis and meiosis. The next question is numerical change in chromosome number which is not exact multiple of haploid genome is triploid, autopolyploid, allopolyploid or aneuploid. So its right answer is aneuploid. While in case of diploid, triploid or tetraploid, these are exact multiples of the haploid genome. As we know that this ploidy is of two types. Euploid, there is a true ploidy. It includes monoploidy, diploidy or polyploidy. Polyploidy is again further of two types, that is autopolyploidy and allopolyploidy, while this aneuploidy, it shows difference from the basic haploid set of chromosomes. Hypoploidy, which is monosomy, means deletion of one chromosome from the diploid set. Then allosomy, that is deletion of two chromosomes from the diploid set. Then we have hyperploidy, that is trisomy, that is having one extra chromosome in addition to diploid set. Then we have tetrasomy, that is containing one additional diploid set of chromosome in addition to the first diploid set of chromosome. So the numerical change in chromosome number which is not exact multiple of haploid genome is called aneuploid. And aneuploids are organisms or cells that have a chromosome number that is not an exact multiple of haploid number. Aneuploids have a partial set of chromosomes due to addition or deletion of specific chromosomes. They are described related to euploid normal conditions with the general form of 2n plus minus 1 or more chromosomes. Examples of aneuploids include nullisomics, that is 2n minus 2, and monosomics, that is 2n minus 1. Then we have trisomics, 2n plus 1, and tetrasomic, 2n plus 2. In contrast, triploids, autopolyploids, and allopolyploids are euploids, meaning their chromosome number is exact multiple of haploid number. So this aneuploid is the right answer. Next question is, if a part of chromosome gets separated and reattached in reverse position in the same chromosome, the mutation is called as, our options are deletion, translocation, inversion, or polyploidy. So its right answer is inversion. Inversion means when some part of chromosome is detached, then it gets reattached, but in reverse position. So that type of mutation is called inversion. We have chromosomal mutations which include inversion, that is separation of chromosome part, then it gets reattached in reverse position. While deletion, that means complete separation of segment of chromosome. Then we have duplication, that means addition of some part of chromosome. Then we have insertion, again, that is addition of some part of chromosome to the another chromosome. Then we have translocation, that is shifting of one part of chromosome to the next chromosome. It is called translocation. So if a part of chromosome gets separated and reattached in reverse position in the same chromosome, the mutation is called inversion. Inversion occurs when a segment of chromosome breaks off, reverses in orientation, and that reattached to the same position on the original chromosome. This type of chromosomal rearrangement does not involve the loss of or gain of genetic material, but rather a reorganization of existing genetic sequences within a same chromosome. Inversion can be classified in two types. It is of two types, that is paracentric inversion and pericentric inversion. Paracentric inversion means which do not involve centromere. So जिसमें centromere involved नहीं होता है, इस तरह के inversion को हम बोलते हैं paracentric inversion. This is the centromere, so this is not involved. If this centromere is involved, then this is called pericentric inversion. If this is involved, this is called pericentric inversion. Inversion can have significant effects on functioning of genes and can lead to formation of abnormal gametes during meiosis, potentially resulting in unbalanced offspring. So in contrast, deletion involves loss of segment of chromosome, and translocation involves exchange of genetic material between non-homologous chromosomes. Then polyploidy refers to the presence of more than two sets of chromosomes in a cell or organism. The next question is, what is true about the law of independent assortment? Our options are applicable to all recessive alleles, not applicable to genes present on the same chromosome, applicable to all dominant alleles, applicable to all genes on the same chromosome. So its right answer is not applicable to genes present on the same chromosome, because there are chances that these genes gets transferred in linked form. So we have this, we can explain this law with the help of this cross that is called dihaprid cross. Here we can take two characters into consideration. One is the seed color, one is the seed shape, and other is the color of cotyledon. So for example, we have this character that is yellow color and round seed. This is the dominating character, and the recessive one that is wrinkled seed and green in color. So these are two parental combinations which are taken into consideration. In addition to this, we get in F2 generation we get two new combinations. One is round green seed and other is wrinkled yellow seed. So these are two combinations, with new combination which arise from these two parental combinations. So this is possible because of the independent assortment, but the genes which are assorted independently are present on different chromosomes. They are not present on the same chromosome. So this law states that alleles of different genes are inherited independently within the organisms that reproduce sexually. This means that the alleles of chromosome 2 or more different genes get sorted into gametes independently of one another. The law of independent assortment is based on the principle that the segregation of alleles for one gene is independent of the segregation of alleles to another gene, as long as the genes are not linked. So this is the condition. So these genes should not be linked, means they should not be present on the same chromosome. However, the law does not apply to genes that are present on the same chromosome, as these genes are linked and do not segregate independently. Linked genes are not. These linked genes are genes that are physically close together on the same chromosome and tends to be inherited together. So ये जो genes हैं ये transfer होती है together. So this is true about this law of independent assortment, that this is not applicable to genes present on the same chromosome. The next question is, tRNA has the function of, our options are carry genetic code to cytoplasm, transferring information to mRNA, transcription, adapter for attaching amino acids. Its right answer is it acts as adapter for attaching amino acids. This is also known as adapter molecule, and it is also known as soluble RNA because it is soluble in a specific concentration of NaCl solution. So that's why this is also known as soluble RNA. This RNA consists in two forms. Transferred tRNA consists of two forms. One is the two-dimensional structure that is known as clover leaf structure, and other one is a three-dimensional structure which is L-shaped structure. And this two-dimensional structure was given by Robert W. Hole in 1965. In addition to this, the crystallized structure of this structure of tRNA was studied by Alexander Rich. Then there is one hypothesis that is known as adapter hypothesis. This adapter hypothesis was given by Francis Crick. If we talk about its structure, it contains the 3' and some specific bases which are cytosine, and adenine bases. This arm is known as amino acid, etc. Then we have this loop, that is T loop. It contains one unusual base that is designated by psi, which is known as pseudouridine. Then we have this D loop. It also contains one unusual base that is called dihydrouridine. Then in between this anticodon loop and T loop, we contain this tRNA contain this variable loop and variable arm, which varies in size and is located between anticodon and T psi C loop. Then we have this anticodon loop. It contains the bases which are complementary to codons which are present on mRNA. So इसके complementary जो bases होते हैं codons के complementary bases which are present on this tRNA, which are known as anticodons, and these are responsible for the selection of particular amino acid. Then we have this D loop. It contains again this D loop and T loop. They act as ribosome binding sites, and they contain unusual bases. This transfer RNA primarily functions as an adapter molecule during protein synthesis, linking the genetic code in mRNA to a specific amino acid. This is the main function of this tRNA. They encode for, so means the message which is conveyed by messenger RNA, and this tRNA helps to bring the amino acid, particular amino acid, according to the codons which are present on mRNA. So the main roles of tRNA include adapter for attaching amino acids. So tRNA can carry specific amino acids at one end and has anticodons on the other end, which pairs with complementary codons of mRNA during translation, then transferring information to mRNA. While tRNA does not directly transfer information to mRNA, but it does play a crucial role in translating information encoded by mRNA into a sequence of amino acids during protein synthesis. So this is the important function, that it acts as a adapter molecule. So this is the role of tRNA. Next question is, a plasmid lives together with chromosome, can replicate independently, cannot replicate, show dependent assortment. So its right answer is can replicate independently. So plasmid is extra chromosomal DNA which is present in certain bacteria that contain genes for antibiotic resistance, and is also known as the fertility factor, designated by F positive, and it acts as vectors in biotechnology. So this can replicate independent of this chromosomal DNA. The next question, bacterial cell consisting of flagella on whole surface are known as, our options are peritrichous, monotrichous, lophotrichous, or amphitrichous. So the right answer to this question is peritrichous. So we have different bacteria on the basis of flagellation pattern. One is the atrichous, means bacteria having no flagella, they lacks flagella. The next we have monotrichous. Here there is single flagella at one end. Then we have lophotrichous. This image is wrong. Lophotrichous means having bunch of flagella at both ends. Then we have another category that is called cephalotrichous. Cephalo means head, so it contains a bunch of flagella at one end. This is called cephalotrichous. Then we have amphitrichous. Amphitrichous means having single flagella at both the ends. Then we have peritrichous. Here the bacterial cell consisting of flagella on whole surface, which are uniformly distributed throughout the surface of bacterial cell. We have classification on the basis of flagella. So first one, monotrichous. Its example is Vibrio cholerae. So bacteria that contains single flagella at one end, that is Vibrio cholerae, and Pseudomonas aeruginosa. Then we have lophotrichous bacteria having bundle of flagella at both ends. So this is lophotrichous. Cephalo means head, so it contains bunch of flagella at one end, and this is meant for the directional movement of the bacterial cell. Then we have amphitrichous bacteria having single flagella at both ends, that is called amphitrichous. It includes the example that is aquaspirillum. Then we have peritrichous bacteria having flagella all over the cell surface. It includes E. coli, Salmonella, and Klebsiella. Then we have atrichous bacteria, means bacteria without flagella. We have example that is Shigella. The next we have classification of bacteria on the basis of its shape. We have different types of bacteria on the basis of shape, like we have spheres, spherical bacteria, rod-shaped bacteria, and spiral bacteria. So the term used for spherical bacteria is coccus for singular, cocci for plural. Then we have single sphere, that is called monococcus. Then we have two spheres, that is diplococci. Then we have staphylococcus. Staphylococcus means having a bunch of cells. Then we have streptococcus, means chain of cells. Its example include diplococcus, example include streptococcus pneumoniae, while this staphylococcus includes staphylococcus aureus. Streptococcus pyrogenes is example of streptococci. Then tetrad. Then we have one condition that is called sarcina. Here it forms a cube-like structure of the spherical bacterial cells, that is called sarcina ventriculi. The next we have rod-like bacteria, which are known as bacilli. So we have chain of bacilli, that is streptobacillus. Then we have flagellated rods, like that is Salmonella typhi or E. coli. Then we have spore formers, that is Clostridium botulinum, which is responsible for food poisoning. This is the bacteria which is responsible for food poisoning. Then we have spiral shapes, that include Vibrio, Spirilla, and Spirochete. This was all about for today's discussion about these questions. I hope you will get some idea from this presentation. If you have any questions, queries, and any suggestions, give it in comment section. Thanks for watching. Have a wonderful day.